All right, hello everyone. So you should see our warm-up problem on the board. We have a image, one of our figures from last time, tracking our alveolar pressure and our intrapleural pressure. So what I'm going to ask you to do is to look at that graph and read a couple things from it, because we're going to continue talking about pressures, and we're going to move hopefully into talking about volumes as well by the end of class. Okay, so now that we've had a second to look at this on our own, let's look at this together. So what we're looking at on the right here is a chart, right, of the pressure in the alveoli and the pressure in that intrapleural space over the course of a full breath. So over inspiration, breathing in, expiration, breathing out. So on our x-axis here, it's really time, and we've shown the time spent during inspiration and the time spent during expiration. And our y-axis here is our relative pressure. When we say relative pressure, we're talking about relative to atmospheric pressure. So we know if we look at just the alveolar curve, right, pressure of the alveoli, we would immediately be able to tell, even without having this label up at the top, that we're starting with inspiration and going into expiration, because we see this sort of sinusoidal curve where the alveolar pressure is negative during this first half, and when we have a negative relative alveolar pressure, negative relative to our atmospheric pressure of 0, so we know that air is going to move down its pressure gradient into the lungs. And so then when we have a positive alveolar pressure, we know that positive means it's more than the atmospheric pressure, which means we know we're breathing out, which is going to give us expiration, right? Air is still just moving down its pressure gradient. But what we're really focusing in on in this diagram is some relationships between the alveolar pressure and the intrapleural pressure, which hopefully you remember, right? Here's the chest, right? Here are some lungs attached to their bronchi. There we go, going up into the chest. So when we're looking at that intrapleural pressure, we're looking at the pressure here between the lungs and the chest wall, filled with intrapleural fluid, but contained within this pleural sac. Okay. So when we look at our diagram, first thing I asked was, which is decreasing more, the alveolar pressure or the interpleural pressure? So to figure that out, right, we might look at first our alveolar pressure, right? See, it goes from 0 to -1 during inspiration, okay? So it goes down 1. Then we might look at our second line for our interpleural pressure, right? We went from -4 to almost -6, right? So looking at this graph, we can see that actually the changes to the chest wall during inspiration that help us breathe in have an even greater effect on the interpleural pressure than they do on the alveoli themselves, which kind of makes sense, right? Because it's this intrapleural pressure that is exerting the force on the alveolar pressure, kind of essentially. Okay. So then our second question is asking whether the intrapleural pressure is increasing or decreasing during expiration. Okay, so we got expiration over here on the right-hand side. Well, you can see it's increasing. Great. Okay. Now we relate this to the transpulmonary pressure. So this is potentially the trickiest thing to read off of this graph, right? The transpulmonary pressure is defined as the alveolar pressure, right, minus the intrapleural pressure. Okay. So it's the difference between these two curves. So this yellow region, this has highlighted what our transpulmonary pressure is going to be. Someone's trying to ask, hold on, let him in. Okay, great. So if we're looking for whether it increases or decreases during expiration, we'd want to look at what it is at the start and then at the end of our expiration. Okay, so at the start, we have this blue line down the middle, right? So if we took our alveolar pressure 0, we're almost down to -6 here. So we'd have a transpulmonary pressure of approximately 6, right? And then at the end of expiration, we have our alveolar pressure of 0. We have our intrapleural pressure of -4. So we would have a transpulmonary pressure of approximately 4. So what we can see by comparing those two numbers is that the transpulmonary pressure has decreased over the course of expiration, which kind of makes sense because this transpulmonary pressure, right, is the pressure on that alveolar wall, right? The pressure across the lung, that pressure to expand the lung. So we can see that at the end of expiration, we have kind of a less of a pull on the wall of that alveolus. If we want to think about the factors responsible for these changes in transpulmonary pressure, basically we would note that the transpulmonary pressure is responding to anything that affects pressure in the alveolus, anything that affects pressure in the interpleural space. So it's really these two factors responsible for the changes in transpulmonary pressure. Okay. So we just went through this in order to kind of practice reading this graph because I thought that this was one of the harder ones in your textbook, but it's really interesting and helpful to look at the relationships between all these pressures and forces that we're tracking, because they're all important to how we actually breathe. Okay. So now we're going to move forward into the factors affecting pulmonary ventilation. So now we're going to start talking about things like compliance and resistance of the lungs, and hopefully we'll get into lung volumes by the end of class, but we'll see how far we get. Okay. So when we're thinking about pulmonary ventilation, right, pulmonary ventilation is how much air you're pulling into the lungs. How much air we pull in and how fast we pull it in is going to be dependent on the lung compliance and the resistance of the airway. And remember, this idea of compliance has to do with the relationship between pressure and volume, right? Like how much volume you can get into something without creating a bunch of pressure, right? We saw that that was one of the differences between our arteries and our veins, right? The veins were floppy, they were highly compliant, meaning they could hold a lot of volume. Now we're going to think about compliance with respect to the lungs. Okay. Airway resistance. We also talked about resistance when we talked about the cardiovascular system, about the vasculature, right? So remember that resistance often has a lot to do with radius, right? The radius of a vessel. So before we were talking about radius of arteries and veins, now we're going to think about radius of airways. So we'll expect to see our old friend smooth muscle again, and a lot of the same factors that affected smooth muscle in the vasculature is going to affect smooth muscle in the airways. Okay. So before we get into that, let's make sure we're awake. Let's do a poll question. So when we were talking about capillaries in the circulatory system, we said they had low resistance because they covered like such a big area, even though any individual capillary was quite small. So what do we think that means in terms of the structure of tubes generally? So what this is saying is that individual tubes in the conducting zone are decreasing in radius, but we still have pretty low resistance in the conducting zone, despite the fact that we have decreasing radius of individual tubes. Okay. So about half of you got this, right? So this is why we added the sort of relationship to the capillaries at the beginning, right? Like the thing about the capillaries was they branched so much, and you have so many capillaries, such big capillary beds, that for overall flow of blood flow, it's like the capillaries are giant. It's like you're pouring blood into a lake, right? Same thing is happening when we're thinking about our respiratory system, that despite the fact that as we're branching, right, from our primary bronchi to our secondary bronchi to our tertiary bronchi, every time we split, even though the tube gets narrower, there are more. So if we were to add together their volume, they would end up having a larger volume, which creates a low resistance for the lung as a whole. Okay. It is true that they do have smooth surface. The conducting zone does have cilia, but the cilia in the conducting zone have to do with the mucus, right? We talked about the mucus escalator getting particles out of the lungs, but the cilia don't really push on gas, right? They don't push on air. They push on actual like solid matter pretty much. Okay. And decreasing radius does affect airflow. It's just not if we're looking at the system as a whole instead of one individual tube. Okay. So before we touch back on resistance, let's make sure that we understand compliance and how this relates to the lungs. Okay. So lung compliance refers to basically how stretchy the lungs are. And if we think about the mechanics of breathing, we can, and hopefully intuitively kind of understand why this would be important, right? When we're breathing in, what we're doing is using our diaphragm and our external intercostals to expand the region in the thorax, expand that volume. We're asking the lungs to follow along with the chest wall and also expand in order to drop alveolar pressure, and basically suck in air towards this area of low pressure. So compliance is kind of a measure of how the lungs can be stretched. So basically, as we change their volume, how much pressure that's kind of creating. So if we wanted to calculate lung compliance, it might look something like this, right? So let's say we have lung compliance, and I like to put in little numbers. When we have equations, I think it's easier to practice them with some numbers to make sure we understand what's going on. So like, let's say we have a lung compliance here. What we're going to hold steady, right? Let's hold steady this difference between the alveolar pressure and the intrapleural pressure, right? We're trying to create some specific transpulmonary pressure, essentially. Okay, so we're going to set that equal to 5, right? So let's say we need this pressure, or we're going to create this difference in pressure of 5. Okay, so then we want to think about the volume here, right? Because lung compliance has to do with the relationship between this pressure across the lung and the volume of the lung. So let's say we have a lung compliance of like 2, right? So we'll make our first sample equation a lung compliance of 2. And we want to know what happens with this change in pressure of 5, right? So you can see here that, and I know I didn't put units anywhere, that's on purpose because we don't want to have to bother with conversions like we do in phys lab if we have real numbers. But this might correspond to, right, like a difference in 10, right? So if we fixed it at 5 as our difference in pressure, we told ourselves this lung compliance is 2, right? So we know this has to be 10. Okay. What if we have a more compliant lung then? So let's see what happens if we have a lung compliance of 5. Let's make this 5, all right? So lung compliance of 5, and we're going to keep that same difference in pressure, right? And part of the reason for keeping that same difference in pressure, right, is that difference in pressure is dependent on the intrapleural pressure, which is created by our muscles, right? It has to do with the alveolar pressure, which has to do with the volume in the alveoli. So these are factors that have to do with physical traits about our body. Okay, so if we have a lung compliance of 5, and we know this pressure difference is going to be 5, right? We can then see that our change in volume here is 25, right? So this, at least to me, I like math. I hope you also like math. I apologize if you don't. But what I'm showing you here is that, like, with a lower lung compliance, right, with this lower lung compliance of 2, we would only get in 10 units of air, right, with a lower compliance. But if we have a higher lung compliance, right, 5 is higher than 2, we got a lot more bang for our buck, right? Now we're getting 25 units of air for that same delta P of 5, right? So a higher compliance means we can get a lot more volume given the same transpulmonary pressure, basically. Okay. So this is kind of what we mean about the ease with which the lungs can be stretched. Ease has to do with this difference in pressure. And we can see that it's easier to get air in to a more compliant lung. If you exert that same amount of pressure, you'll get a lot more air. So you don't have to memorize this equation, but I think it's helpful for understanding compliance and how differences in compliance are related to differences in volume and pressure. Okay. The larger lung compliance makes it easier to inspire, easy to breathe. And so a small change in that transpulmonary pressure, which is basically this bottom thing, right, that I had set equal to 5 in my example equations, right, a small change in that with a highly compliant lung would make a big difference for volume. So there are factors that affect the compliance of lungs, and there are some diseases that affect the compliance of lungs, which is why we care. So one thing is that a more elastic lung is less compliant. Elasticity has to do with kind of like storing pressure. So if you're storing a bunch of pressure there, it basically makes the lung less compliant. Like we remember when we were thinking about our arteries and our veins, We think of the arteries as very elastic as pressure reservoirs, our veins as volume reservoirs being very compliant. This is the same relationship, just showing back up in the lungs. If you have a very elastic lung, it won't be very compliant. The unique kind of factor that we have in the lungs that relates to compliance is something called surface tension. So the surface tension in the lungs, we're thinking about the inside of an alveolus, right? So I'm going to draw one quick for us. I'm sure there's a drawing on a later slide, but I don't like to have slides that are all words. Okay, so we're thinking about surface tension. So we're thinking about an alveolus. The surface tension is due to the fact that the inside of the alveolus, well, it does contain air, right? These membranes are wet. So basically they have a thin layer of fluid lining the alveolus. So let's pretend these are water molecules, right? When you breathe, the air is, maybe a little humid, maybe a lot humid, but regardless, there's going to be water vapor in it. So this makes sense, right? So we've got a fluid layer lining the alveoli. Surface tension, right, is really due to the fact that water molecules are attracted to each other. So hopefully you vaguely remember from your earlier bio classes or your chemistry classes, this idea that water molecules, they try to form hydrogen bonds between them, right? And that's what surface tension is, right? Is water molecules forming hydrogen bonds, which means that they're also kind of exerting a pull across the alveolus, right? They're trying to collapse into a single droplet, which means that there is a force trying to pull them all together, pulling inward on that alveolus, basically creating a force for that alveolus to compress and contract, or sorry, to compress and collapse. Right, so that's due to the attractions between these water molecules. Okay. So if we have greater tension, so basically what we're saying here, right, is if this force, right, of these water molecules trying to form a droplet in the center, pulling the walls of the alveolus inward, if this force gets bigger, it makes the lung less compliant because there's basically more pressure that we have to fight against in order to expand the alveolus in order to increase its volume. Okay, so this surface tension of the fluid inside the alveoli really does affect how easy or hard it is to expand the alveoli. We don't want that. We don't want our alveoli to collapse. We want to expand them. We want it to be easy to put air in there because we want to do gas exchange. So in order to combat the surface tension between water molecules in the alveoli, just in that layer that's touching the wall of the alveolus, we secrete something called surfactant. So way back at the beginning of the unit, we were looking at some of our cell types in the alveoli, and we looked at the type 1 cells as being basically forming a barrier to gas exchange along with those endothelial cells in the capillary, so that we had this sort of a double layer of cells, which is later on going to be the two layers of cells that we'll have to see things crossing in order to get into the blood. So those were our type 1 alveolar cells. We're forming that basically epithelial layer. Our type 2 cells are not a discrete layer, they're interspersed throughout. Their job is to secrete surfactant. So what surfactant is it's basically a detergent and it decreases the surface tension. So when we're looking at that alveolus that I had drawn, right? So we have our alveolus, all right? I'm going to draw us just a couple water molecules, right? The water molecules trying to hydrogen bond with each other, so they're creating forces inward, right? So they're creating forces trying to get close to each other. I'll draw a couple more of them. Okay, so those are our water molecules. This is showing our tension. What the surfactant does is the surfactant basically comes in and breaks it up, right? So surfactant is also fluid, but it's not one that forms hydrogen bonds with water molecules. So it kind of blocks their interactions with each other. So it fights against the creation of this surface tension. It gets in the way. And this means that surfactant has the effect of increasing lung compliance, because if we have surfactant in there, right, we can erase these arrows, right? Surfactant means that these water molecules can't see each other as well, basically. So we'll still have some tension forming. So they have a little bit of a force pulling them together, but because they have a lot of surfactant here, this force is less, which means that the lung compliance is greater. Basically, it gets easier to expand this volume again, because this surface tension is a force fighting against the expansion of the alveolus. Great. So this takes us to a more professional drawing of what's going on. Okay, so here we're looking at two alveoli. So we have two alveoli. We can see that they've shown us the force from the surface tension inward towards the center of the alveolus for these two alveoli, which have two separate pressures, P1 and P2 here. Okay. So a thing to think about as we're thinking about this surface tension, right? If we let this blue arrow I circle do its job, right? If we allowed this alveolus to follow this tension created by the surface tension and allowed it to get smaller, we know that when a container gets smaller, right, that raises the pressure. We remember that inverse relationship between pressure and volume. So basically, this surface tension, if we allowed it to work, would make this P1, the air inside the alveolus, get higher and higher pressure, right, as the alveolus 1 got smaller in response to the surface tension. Now, second thing we know is that pressure gradients create flow, right? So if we're letting alveolus 1 get smaller and we're raising the pressure in that alveolus, the air might just want to flow into alveolus 2, right? If alveolus 2 isn't at that higher pressure, right, we might end up with all the air flowing into the bigger alveoli, and then eventually just ending up with like all our air just kind of concentrated in the biggest alveoli, which is not efficient for gas exchange. We don't want that to happen. We do want air to end up in all the alveoli. So in order to do that, your body is going to try to make it so that this P1, right, and this P2 and the pressure in every alveolus is the same, so that we don't have air totally leaving some of our other alveoli. So that's going to be one of the roles of surfactant. Okay. So now I can use this equation at the top here. So the pressure here in the alveolus, so like the P1, is equivalent to 2 times the tension divided by the radius. I just recopied that. Okay. Going to go to our next drawing. Does it click on? Yeah, great. Okay. So what we can do is we can change, we can, what did I want to say? I lost my train of thought. We can change the amount of surfactant in our different alveoli in order to make sure that this equation basically balances. That's what I wanted to say. So you'll note here, essentially, our surfactant molecules are these squiggly guys, right? They might kind of look like phospholipids, right? But they're surfactant, right? So we can see that there's a lot more surfactant here in the smaller alveolus than in the larger one. And the reason for this is that we're going to have to combat this effect of radius. So T here has to do with the surface tension. So we need, in order to have an equivalent pressure, an equivalent P1 and P2, as the radius goes down, or sorry, yeah, we need to have a trade-off here, basically. We'll do a practice equation with this later. I had planned one vaguely, but I don't have the slide up. I forgot to add it in. Okay. But basically, surfactant molecules are fighting against this tension. We need lots of them in small alveoli because otherwise the pressure would get too high. Right. So moving forward to resistance. Okay, so and I will make a note that we actually do go through the practice equations for surfactant later. Okay, let's talk about resistance. Okay, so as we know, when we have the diameter or radius of a tube decrease, that increases the resistance through that individual tube. But when we're thinking about our airways in total, while they are getting smaller in diameter as they branch, they're also increasing in number. Okay. We do need a pressure gradient for air to flow through the lungs. And the resistance is usually kept low so that that pressure gradient doesn't have to be too great. If we have increases in resistance, it's going to make it harder to breathe. Okay. So we're going to take a look at some ways we can change that resistance and some things that might increase that resistance. So here what we're looking at, on the bottom, we're looking at the breath volume, volume of air moved into and out of the lungs. So increasing during inspiration, decreasing in expiration because the air is moving in and out. On the top image, we're looking at two curves. So we're looking at the intraalveolar pressure. The intraalveolar pressure, and we're looking at how much pressure we need to create that specific breath volume with normal resistance. So that's our dark blue line. So here is our normal resistance line. Right, so we get to a pressure of about -1 during inspiration, pressure of about +1 during expiration, right? That's with normal resistance. But because we know that flow is basically our pressure gradient divided by resistance, if we increase resistance, we have to also increase our pressure. So we see that if we have a situation of increased resistance, we need to drop the intraalveolar pressure down much lower to get that same breath volume, to get that same flow. And then it has to get much higher with this increased resistance, again, to get that same breath volume, to get that same flow. Okay. So an increased resistance, this curve is basically going to have greater amplitude here. So we have a number of different forces or factors that affect the airway resistance. Two of them we refer to as passive forces. So for our passive forces, we basically have the force of the transpulmonary pressure and then the tractive forces. So I want us to remember here, stepping back for a second, when we're talking about airway resistance, we're not talking about the individual alveoli anymore. Now we're talking about like the bronchioles. Okay, so we're talking about the tubes now instead of the little sacs. So this is very slightly different, but it's important to keep in mind. Right. So passive forces on those airways, one we've talked about when we were talking about the alveoli. So the transpulmonary pressure, so that pressure created by that intrapleural space, has an effect on the alveoli, also has an effect on the airway, so also has an effect on the tubes. So when we're inspiring, when we're breathing in, the transpulmonary pressure is increasing, right, as our chest wall pulls away from the lungs, right? So that caused our alveoli to expand. It also causes our airways to expand. So we're going to end up decreasing the resistance in those airways as the transpulmonary pressure increases as we're breathing in. Our other force, our other passive force that's going to be happening is something called tractive forces. So tractive forces is the actual physical pull of the tissues. So this isn't like a pressure anymore. This isn't PIP. This isn't one of the pulmonary pressures we talked about. This is talking about like the fact that we have literal connective tissue connected to those airways, right? There's flesh touching them and they pull on the airways. So as the tissue moves away from the airways, right? Like as your physical ribs expand, they physically pull on the airways due to some connections there. And those are your tractive forces. Okay. So those are our passive forces that affect airway resistance. And both of them are affecting resistance just in the way we talked about when we talked about with vasculature, right? They're affecting the radius and therefore affecting the resistance. So just like with the vasculature, right? If that radius gets bigger, right? If we dilate our vessels, if we make them wider, it's going to make it easier for things to flow through, right? So we have decreases in resistance as we dilate. We have increases in resistance as we get smaller, as that radius gets smaller. Okay. Our two other forces, other than the passive forces that we want to think about when we're thinking about airway resistance, are the contractile activity of smooth muscle and mucus secretion. We're going to go through those next. So when we're thinking about the smooth muscle, when we're thinking about airway resistance, we're specifically thinking about smooth muscle surrounding our bronchioles. So bronchioles have lots of smooth muscle, which contracts and dilates much in the same way as our vasculature did. So when we have bronchoconstriction, so just like our vasoconstriction, right? With bronchoconstriction, we're going to see the smooth muscle contract, which means the radius decreases, which means that our resistance increases. Right? Bronchodilation, smooth muscle relaxes, radius increases, resistance decreases. Right? So the same sort of physical relationship between the radius and the resistance exists in the bronchioles just like it did in the vasculature. Okay. The smooth muscle in the bronchioles is also under similar control as the vasculature was. So we're going to see that we have extrinsic control, like hormones and nerves. We're going to have intrinsic control, like levels of carbon dioxide and oxygen, the same way we did when we were looking at the vasculature. Okay, so having said that, let's take a look at this next question. So what do we think some specific examples would be? Okay, so thinking back to those extrinsic and intrinsic factors, which we're comparing now in the bronchioles compared to like our vasculature. Okay. So our extrinsic factor might be something like epinephrine, right? So epinephrine, remember, is a hormone, but also related to the sympathetic nervous system, right? Sympathetics, when they get to the adrenal glands let out epinephrine. Intrinsic here is carbon dioxide. So it's like when we were talking about our active and reactive hyperemia, right? When we were talking about how blood vessels respond to the metabolic activity of tissues and the buildup of oxygen or carbon dioxide, that was an intrinsic factor affecting smooth muscle in our vasculature. It's still an intrinsic factor when we have carbon dioxide affecting smooth muscle in our lungs. But these other factors do come into play. It's just here's how we would define them. So our extrinsic control is largely going to be under control of the autonomic nervous system. So when we were thinking about vasculature, we mostly did just think about the sympathetics, but the parasympathetics do have a really big role here in the airway resistance as well. Okay, so our autonomic nervous system, remember it's involuntary. We have our sympathetics, we have our parasympathetics. Sympathetics in the case of our airways are going to create relaxation of smooth muscle and therefore bronchodilation. And I'm saying this so slowly because remember, this is kind of the opposite of what we had happening in the vasculature, right? So it has a different effect here on our smooth muscle in the airways than the smooth muscle in the vasculature. So the sympathetics are going to do bronchodilation. So they're going to dilate these bronchioles. They're going to make it easier for air to get into your lungs faster, essentially under sympathetic control. The parasympathetics are now going to have the reverse action. So the parasympathetics are rest and digest, right? Rest and digest, in the case of the lungs, means that you can get air a little more slowly. So the parasympathetics are going to cause contraction of the smooth muscle and therefore bronchoconstriction, increasing resistance, making it a little harder to breathe, making it take a little longer, right? So the effects of sympathetics and parasympathetics on muscle really depend on what system we're talking about. And when we're thinking about now the respiratory system, we see different effects than we did when we were thinking about some of our vasculature. Okay. So the extrinsic control of the bronchiole radius is also under hormonal control. But the main thing we want to think about is epinephrine, which is kind of halfway between thinking about sympathetics and thinking about hormones. Causes relaxation of smooth muscle, causes bronchodilation. When we're thinking about intrinsic control of bronchiole radius, we have some new kind of factors coming into play. So one thing that we have happening is that we have histamine creating bronchoconstriction. So histamine gets released during asthma and during allergic reactions. And it actually causes the airways to constrict and therefore resistance to increase, making it harder to breathe. And histamine also increases mucus secretion, also basically making it harder to breathe. CO2, however, causes bronchodilation. Right? So CO2, a buildup of CO2, means that we want to do more air exchange. So that's going to cause bronchodilation, increasing airflow, making it easier to get air flowing so that you can exchange that carbon dioxide for oxygen. So histamine and CO2 would be acting on a pretty local level. So they'd be coming from a very nearby location to that bronchiolar smooth muscle, whereas these extrinsic controls would be coming from farther away. Okay. So largely when we think about airway resistance with respect to the respiratory system, we're interested in pathology and we're interested in diseases. So I know I got some questions about this in lab sometimes. So What we're looking at here first is a picture of a bronchospasm. So bronchospasms are really what happen during asthma attacks. So asthma is caused by spastic contractions of the bronchiolar smooth muscle, which is to say, like, not on purpose, right? Like not for a good reason, like kind of like a cramp almost in the bronchiolar smooth muscle. So we can see our picture on the left there. We have a normal airway, looks nice and expanded, nice and dilated. That smooth muscle is nice and happy. We have an open airway. But in an asthmatic airway, we have two things happening. So one thing, that middle picture is an asthmatic airway in its relaxed state. So we can already see that those relaxed smooth muscles already look a bit chunkier, right? They've already kind of compressed to that airway a little bit, right? Those look like some strong smooth muscle cells, right? And then on the right, during the asthmatic airway, during the attack, when they tighten up, we can see that the airway gets really small, right? The radius gets really small, resistance gets really high. And we can actually see labeled on the alveolar sac there, right, is that this isn't just affecting the ability of air to come into the lungs, it's also trapping air in those alveoli, right? Because air has to move into the lungs through the airway and then out of the lungs through that airway. So we're not just having a problem breathing in, we are in total having an airway problem moving air through that airway at all. So we can also end up with air kind of stuck in the lungs. Another time we think about diseases that create problems with airway resistance are places like chronic obstructive pulmonary diseases. So you may have heard of COPD. It's a common lung disease. This also is affecting the resistance of airways, and so the ability to efficiently move air through the tubes into and out of the lungs. And when we take a look at our lung volumes, we'll come back to this and think a little bit more about COPD as well. So, respiratory volumes and airflows. So we want to know something about lung volumes and capacities. We want to know something about how we test pulmonary function. And we want to get to this idea of alveolar ventilation. We're definitely not doing all of this in the last minute of class, but I do want to just quick intro this idea of lung volumes and capacities. Okay. So this is an old school diagram of a spirometer. So a spirometer is something that measures basically lung volumes. And we're going to name different parts of your lung capacity based on basically what part of the breath they're part of. So our first lung volume we think about is something called the tidal volume. So that's a single unforced breath. If you're just quietly breathing normal, the amount you're breathing in when you're not thinking about breathing, that's your tidal volume. Yeah, and we're at time. We'll go over these other volumes next week as well. So you will have a quiz for this weekend. I didn't put it up yet because I wanted to see how far we would get. But stay tuned, and I'll send out an email once that's up this afternoon.
I have a quick question about the slides from last meeting.
Mm-hmm.